Good morning, everybody. Thank you for coming to the third annual Synapse Roundtable. My name is Michael Graglia. I am the Managing Director of the Syngap Research Fund, and it is such a thrill to welcome our incredible speaker here today.
I am delighted to introduce Dr. Chung. She is a clinical and molecular geneticist at Columbia University. She's one of the world's experts in the genetic basis of human diseases. She's also the director of clinical research at Simon's Foundation Autism Research Initiative. She's the PI of SPARK and Simon's Searchlight, which both aim to understand genetic neurodevelopmental conditions. Thank you so much for joining us, Dr. Chung.
Thanks, Geraldine. I'm really thrilled to be here. Let me see if I can share my screen the right way. And hopefully you can see everything. So I'm very pleased to be here, and I'm going to show you hopefully some data that I hope many people can use, and always looking for input on how we can do better. This is part of the umbrella program that I'm calling Simon's Searchlight, although as Geraldine said, we also sponsor SPARK, which is a study that I think of as feeding into Simon's Searchlight. So Simon's Searchlight is predicated on the fact that individuals with neurodevelopmental disorders are clearly not the same, and we can start to understand better, and we must in terms of getting to treatment, subdivide by genetic basis, but also realize that as we subdivide, we can see synergies, and obviously the synaptopathy are a great example of that. This is just showing you the current genes and copy number variants that we include within Simon's Searchlight to say that the knowledge is still increasing in terms of the genes that are involved. And so we continue to expand our genetic conditions that are included within that. And I'll also point out we continue to iterate in terms of inclusiveness. And one of the things we've been doing is increasing the number of languages that we can collect and distribute data in, both in terms of just online writings as well as the languages that our research team speaks. With this, the goal in terms of Simon's Searchlight is again to be able to make for participants, i.e. families, make it relatively easy to participate and make it scalable in terms of the distribution of the information so that we can all learn faster. And as we're doing this, I'll show you some dimensions. We've been thinking especially about how to get more resources than just data to researchers, and I'll get into some of the specifics of that, but I can really feel a shift in terms of the impact that's having on the field. As we're doing this, the low-hanging fruit, so to speak, are to be able to get online data. The reason we've done that, some of you know that as we started this out as something we called Simon's VIP at the time, it was more burdensome on families. We had them come in person, do a couple days in terms of evaluation. We still have those data freely available to researchers, but we realized that we couldn't include as many people due to travel restrictions, especially with COVID. The online community afforded us much more flexibility to understand in real time how COVID was impacting our community, as well as being able to continue data collection. So as we've done that, number one, we collect the data online as well as with genetic counselors who review and be able to really, I think, help us in terms of getting really clean, robust, valid data, having genetic test results from families that we can validate their diagnosis, sharing that information back with families and community groups, as well as making that freely available to researchers around the world. And as we're doing this, as I said, we hope that this is partially powering readiness for clinical trials, as well as being able to power the researchers who are going to develop those understandings of basic mechanism, as well as some of those treatments. This is just an example of the online measures that we have, and the medical history interviews that we do. The point, or one point to this, is that this is not one and done, but this is longitudinal, appreciating that things change over the life course, challenges change as people evolve, and so we continue to collect data as individuals are changing over that life course, both from a medical point of view as well as a developmental point of view. Again, I'll show it at the end, but all of these data freely available. As we're doing it, we want to make sure that this is as I said, available to researchers, but also that this is more short-term affecting clinical care and clinical guidelines. And one relatively easy way to do that we found is through gene reviews, being able to, that's for those of you who don't know, it's an online resource, again, open access, formatted in a consistent way where a busy practitioner can be able to log in for the gene of interest and find sort of clinical care, what they should be doing in terms of health surveillance or interventions. And And again, something that we as genetic professionals continue to update. We take that as a serious responsibility. So that's something that we've powered many of our Searchlight genes to be able to enable gene reviews. I and my teams have done some of those reviews, but many others in the community have done that as well. And then also making sure the variant level data is in ClinVar. So again, it's without any identifiers in terms of participant identifiers, but making sure that diagnostic laboratories have free access to the information so that more individuals get correctly diagnosed as they're going through diagnostic testing. I will say all of this in the very careful curation of those genetic test results we found to be incredibly valuable, although about 90% of the individuals who come into the Searchlight portal, once we review their genetic test results, about 90% of them in fact have the right diagnosis, but there are a good 10% where that's not definitive based on the testing we've had so far. One of the other things Searchlight does is to actually pay on our dime for the genetic testing, for instance, of parents, if they have not been part of that genetic testing process, to make sure that we know whether this was an inherited or de novo variant. That also helps in terms of interpretation of that variant, as well as getting additional information on additional people that may be valuable. And again, that's value added in terms of what Searchlight can do. All of these data available, again, the URL is right here in terms of Safari Base. We have incredible data navigators to also be able to help researchers understand the data in addition to data release notes and data dictionaries. But this is regarding specific clinical data, as I said, genetic data, but also importantly biospecimens. So whether they're DNA samples, induced pluripotential lines, other types of cell lines, making sure that's available. And importantly, an opportunity that we have called Research Match. So this is an opportunity for us to be able to advertise to the community by gene or by groups of genes, research studies that are IRB approved through investigators around the world, to invite them to be part of their research, whether it's online, whether it's in person, but be able to match those researchers with individuals in those gene communities. And that's been, I think, is going to continue to be a very effective way to allow families to participate, not just in Searchlight, of course, but much farther. This has been an important resource that we've been creating, induced pluripotential stem cells. I'm listing here and showing some of the conditions that we've added recently in green, the new cell lines. These are all done again at the expense of the Simons Foundation and freely available both to basic science researchers in universities as well as in commercial settings. Within this, in some cases, we have coisogenic corrected lines so that I think they have both good controls as well as allelic spectrum so that we have many different variants within the same gene, so that if there are any allele specific either differences in mechanism or differences in severity, those can in fact be studied by researchers. So again, all of these freely available. We've had a call out to the community to vote democratically on what lines are necessary or helpful to the community, and always looking again for feedback if there are lines that we don't yet have established. Within this, I started to describe that this research match opportunity is something that's always ongoing and active. And so this is something, again, that we can take literally as studies are ready, whether these are clinical trials or survey-based studies or looking for clinical readiness or a particular measure, whether it be sleep or something else, to help you with your studies. And again, to be able to do it across conditions, which might be helpful in terms of looking across synaptopathies or synaptopathies versus other types of genes. Data sharing really is something we believe very, very strongly in. Again, we, just to be clear, this is all de-identified individuals. We try and make sure that they are in fact qualified researchers by requiring a research distribution agreement, which says that they agree not to try and identify individuals within these studies. And as we do this organically, it grows so that data, for instance, from research match studies organically comes back into Safari Base, so participants don't have to keep answering the same questions over and over again, but can be able to do something once and have many, many people be able to benefit from that. As we're doing this, we do try and cooperate with other groups that are also collecting data. In particular, Citizen, for instance, has been focusing a lot on EHR data, being able to work together so that we can increase the power of all of the different data sources that are available. With this, as I was saying, one of the advantages perhaps compared to some registries that might be individually focused on a gene or a group of genes is there are important differences that at least I've noticed in comparing across our different conditions. individuals, for instance, who may be wanting to explore a particular phenotype, it's very easy to be able to look across genes and be able to see what are the particular genetic conditions that might be associated with either very little of a particular symptom or a high frequency of that symptom. And that might be useful in terms of comparison across those to understand that biology or behavior better. In particular, for the synaptopathies, as we're talking about, we do have very, very rich data collections on some of these groups. I give a lot of credit to the family group leaders. It's in large part because of their efforts to rally the troops and to emphasize the importance of research to their communities. And I won't go through exactly all of the numbers of individuals as of today, but we do have large data sets for all of the ones that I've boxed and shown and might be of interest to the group here. In particular, and not surprisingly, all of them are associated with neurodevelopmental conditions. But in particular, if we look at some of the major sort of conditions that we see, everyone has some degree of cognitive neurodevelopmental issues. Most have issues with epilepsy or seizures, and autism or ASD on the left is seen in a large number, but certainly not everyone. And I want to be clear that although Safari, I think, is known for being an autism sort of umbrella in terms of the research, this is really a genetics first approach in which clearly not everyone within these genetic groups has autism, nor are we solely interested in autism as a manifestation. So in fact, we fund many researchers who study these conditions not solely for the purpose of autism, but for epilepsy or other behavioral issues. The types of things we give back to family are these research snapshots that I'm showing you here. We try and do this as regularly as we can, and especially as we have new data releases. So you can see, for instance, with STX-BP1 in the last data release we had of 68 individuals, or Syngap1 with 31 individuals, being able to see the types of things, the types of distributions of frequency of manifestations that we see. And again, we hope that this information is valuable to families and will continue to enrich it as we think about the numbers increasing, responses to certain medications or problems with certain medications, but deeper detail that we often hear families wanting to have answers to. Within this, we see many different manifestations beyond what I just said, some that are more observable directly by clinical observation, and so getting increasingly deep in terms of some of those assessments, but again, being able to see some things that are common features across many of the synaptopathies, and in fact, across many of the neurodevelopmental conditions that we study. So with that, the other thing that I'll say is that this last year, some of the investigators know we had a request for applications, or an RFA at Safari, that was specifically giving funding opportunities to investigators, not solely to use Simon Searchlight data, but to be able to go deep. In particular, it was genetically focused, so to go deep in terms of understanding mechanism, but importantly, driving us closer to therapeutics. And so I think we've just been able to finish the review of those RFA or those grant submissions, and in fact awarded those applications out. And I'm very excited to see that we are, I think, going to be able to give significant amounts of dollars at the Simons Foundation. I think based on the success this last year, we're thinking this is going to be something to continue doing, to be able to catalyze on the therapeutic side, but specifically for the conditions that we have under Searchlight. So I hope, as a brief summary, this again is something in terms of what might be a resource available from the Simons Foundation. Again, the URL to be able to get any of the cell lines or any of the data that we have here. We have scientists on the Safari team that can help you in terms of, again, any taking requests for anything else that you'd like to see that we don't already have, as well as RFAs that we have throughout the year for funding for any of these programs. So I'll stop there, but thanks again for including me.
Thank you so much, Dr. Chung. It's a real honor to have you in this event. We have a couple minutes for questions. Like Dr. Smith-Hicks, you've actually left some time, which is amazing. Until questions roll in, I just want to say that SRF, I'm sure my colleagues at STX-BP1 and Shank3, we do encourage our patients to take part in Simon Searchlight. And I'm particularly thrilled to know that through partnership with your incredible team, Jennifer and others, we have also been working on getting a connection with Citizen, who's going to talk next. So hopefully we'll be able to get our community taking advantage of both of Citizen's medical records and of all of the incredible investment that Simons does. I just can't say enough I'm so grateful and honored to have you here. If there are no questions about, wait, here they come.
So the first question, in terms of a significant reason why Syngap 1 has half the pool of patients in Safari base as the other genetic difficulty, we support everyone, so we're not filtering anyone. It's simply who volunteers. So if anyone would like to get the word out, certainly we encourage you to, and we continue to expand in terms of capacity based on how many families we have. So you keep them coming, and I promise we'll keep up with you. So I'll give you that challenge. And then, do we have resources for families as far as understanding their disorder, genetics, biology? The answer is yes, absolutely. So we support family meetings, both online, it's been during COVID times, but also in person. Oftentimes we'll have sessions like Genetics 101, or we've had sessions that I've done about gene and cell therapy and how to be able to understand that. So there are lots of resources, and we've recorded all of these. So they're enduring resources that can be available to families asynchronously anytime, and especially I did that because I know families are newly diagnosed every day, and we want them to get that information right away when they're in that state of panic and trying to learn as much as they can. So we can certainly follow up, but they're scattered in a few different places, but YouTube is an easy way to find a lot of the information content, and we're happy to share that as a resource afterwards.
And I will also say, Pat Breland, because you're one of ours, we are working to correct that. There are incredible opportunities from the Simons Foundation, like the cell lines. And I will also mention that the Simons Foundation supports Autism BrainNet, who SRF is a proud partner of. I know it's a horrible thing to think about, but we encourage our families to consider donating their loved ones' brains after they pass. Hopefully we don't have to face that for a while, but it's important for research. So Simons just does so much, and we want to see our patients in that. And hopefully by building this bridge with Citizen, who we'll hear from next, and Simon Searchlight, we'll be able to up those numbers and really take advantage of the generosity of that foundation.
Mike, just one more, since you mentioned Autism BrainNet, an unabashed advertisement, if any of you can get the word out to scientists. We will be having, or actually just released, a request for applications for scientists to use the resource from Autism BrainNet. So again, we're not just creating those resources, but we're giving funds to scientists to do the science. So please encourage them to apply. We'd love to have them use it for your groups.
Absolutely, thank you.